on your own. And if you recall, when you signed up for this, there was some notes about making sure that Java works on your computer. So if you want to try to follow along on your computer and you've already downloaded GSCA and GeneE, you're welcome to do so. But I'm going to do it live and also have screenshots in case me doing it live doesn't work for some reason. Yeah. So here's the problem, right? You have a list of shRNAs, and I'm not going to talk about how you go from all your various conditions, low drug, high drug, time point 1, time point 2, this, that, the other thing, how you get that down to a number. But that's where I'm going to start. The idea that you've already done normalization, subtractions, figure this out, figure that out. What do you think the relevant number for your perturbation is, right? So how do we appropriate? So the question to start with is first, how do we appropriately map shRNAs to genes? As I mentioned, we have, when you do a screen, you'll use 5 to 10 hairpins per gene. So how do you know which gene scored? You know which shRNA scored, but shRNAs have off-target effects. And we're going to talk a little bit more about that in a bit, but absolutely, shRNAs have off-target effects. So you want to know what were the genes involved in my phenotype. So how do we maximize the on-target effect, and how do we map shRNAs to target genes? So again, if you want to follow along, you can go to this website, www.broadeinst.org/tilde, and then my name, and you can download. Is anyone actually going to follow along, just so I don't? Okay, some people are. Okay, good. So if you go to this website, there are 3 files on here. The only one you actually need to download is this final one, vwscreendata.txt. Okay, and I clicked on it to open it, but you can download it. And this is what you get at the end of a screen, right? You've got a perturbation, a gene symbol that this thing was attempting to target, and then some score. Now, obviously, the Excel spreadsheet that came before this had lots and lots of columns of lots of treatments and time points and whatnot. But at the end, this is how this screen in particular, was scored, a number. So what do we do with that? Okay. So one way that we like to recommend, and I should also point out that throughout this analysis, there are many, many, many ways of doing what I'm going to show. The reasons I'm showing the way I'm showing is that these were all developed at the BroadE. It's the ones that we use routinely. I'm not suggesting that they are the only way to map shRNAs to genes or to do network analysis. Not even remotely. There's lots of other ways to do it. This is just one way to get started. And it's a fine way to get started. But there are many ways out there. And I'm not saying that this is the only way you should do it or even the best way to do it. You'll certainly get differences of opinion on that. So the first thing you want to do is open up the GeneE software. That's my daughter. She's cute. Now, when you open up GeneE, okay, Java should start running. Java has been very problematic on Macs of late. If you haven't updated your Java in a while, that could be problematic. OK, I don't want to load an old session. So the first thing you want to do is open a file. You don't want to import, because import kind of restricts the types of files you can load. You just want to open the file. And you want to open the file that we just downloaded. Now, here's another little quirk of GeneE. Sometimes when you go to open a file, you'll just get, at least on Macs, you'll get this spinny wheel down here and nothing's loading. It's like, wait, why is it not popping up? So you just wait a second, cancel, file, open file again, and now everything shows up. So something in there is just not popping up. But so you do that. Let's see, where did I put it? BroadE? Oh, no. Test files, BroadE. Okay, so now spinny wheel again, wait another second, open again, test files, BroadE, and there they are. Okay, so we want to load in this plain text file, which again, it's just a symbol, sequence, symbol, and score. So the most basic kind of shRNA data you can get. When you import, you click, and it tells you this, click the table cell containing the first data row and column. So you click right here on the score, and it nicely highlights what's metadata and then what's actual data. So you click on that, click OK, and it imports it. And you get something like this. Now, I should point out that GeneE is an incredibly powerful program developed by Josh Gould, who sits up on the 4th floor, very nice guy. It does a ton of things. I'm showing you about 0.1% of what GeneE can do. GeneA can do a lot of things, especially it was developed largely to look at microarray data. It can do a ton of stuff. I'm just showing you one very tiny tool that GeneA has. It's a program that has a whole host of other other functions. It could be a BroadE seminar in and of itself. And there's lots of resources to learn about BroadE, or to learn about GeneA. But what we want to do is go to Tools and click on this tool, Rigor. Okay? Now, when you click on Rigor, you can do class comparisons. I'm not going to show that because here we've already figured out the score for our perturbation. But if you wanted to do some of the calculations in GeneA instead of doing them in Excel, you certainly can. I kind of like to go to GeneA only once I've done all the calculations in Excel, just to make my life a little easier. I'm more comfortable working in Excel. But the first thing you want to click is you want to scroll down a little bit and click on hairpins are pre-scored. And you'll notice that when you click on hairpins are pre-scored, all this class comparison stuff grays out. Okay, so you're telling GeneA that you've already figured out the score for your hairpins. Then there's a few other options here. There's a number of permutations. Leaving that at 1,000 is fine. You can increase it if you want to do more permutations, but 1,000 is generally fine. The method to convert hairpins to genes. So this is a big question, right? If you have 6 shRNAs all targeting the same gene, how do you vote for how well that gene scored? One of the simplest ways to do it, and what Rigor will do, you definitely don't want to use the KS statistic. It's not particularly useful for shRNA analysis. You want to select either weighted sum or second best rank. Second best rank does exactly what it says. It will re-rank your hit list based on the second hairpin that scores for each gene. Okay, so by definition, you had to have 2 hairpins scoring for a gene in order for it to be at the top of the list. Weighted sum does more or less the same thing, but instead of going only by the second best hairpin, it weights the best hairpin for a gene and the second best hairpin for a gene. I prefer weighted sum. The hit list that you get from using second best or weighted sum are very similar. So for this purpose, we'll run it with weighted sum. Likewise, gene rank order, positive to negative or negative to positive. In our case, because I'm telling you, the hits, the side of the screen we were interested in were the most positive values, but sometimes they'll be the most negative values. So you can just tell Rigor, are your hits positive to negative, or are they negative to positive? In our case, it's positive to negative. Again, meaning the hits just had the higher numbers or the most positive numbers. Random seed used to generate permutations, don't need to touch that. Adjust gene scores to accommodate variations in hairpin set size. That you absolutely want to have checked off. The reason for that is we have some genes that have 5 hairpins. Some genes, small number, but some genes have 20 hairpins in the library. Clearly, if you didn't correct for the number of hairpins targeting a gene, the gene that has 20 hairpins just by chance is more likely to have 2 that are at the top of your distribution rather than a gene that has only 5 hairpins. So if you keep that checked, then the statistics that come out at the end will be corrected for the number of hairpins per gene. The hairpin ID you can leave as hairpin sequence, and you want to convert hairpins to symbol, okay? Meaning that that's what you want to do. You have 5 different hairpins, but they all share in common a particular gene symbol, so you'll leave that as symbol. Okay? Then we click OK, and it runs, and this does not take very long at all. And what happens is you now get another tab popping up over here. So here's your original data in this tab, but now you got a new tab popping up. Okay? Now, in and of itself, you can't do much here. So what you want to do is if you're on a PC, you can right-click on the tab label, or if you're on a Mac, you can hold down Control and click. Okay? And that pops up this little tab. Not the most intuitive way to access this, but it pops up this tab, and you want to download, and you want to pop up the gene report. Okay, so I do that, and this is your gene report. This is how Rigor has taken those hairpins and said, here's how well these genes are scoring. Now, you can look at it in GeneA, although it's a little harder and you want to do some ranking. So usually the first thing I do is I save it to an Excel spreadsheet. So you can do that by clicking on the save icon up here and giving it some name. So we'll do test.txt. We'll save it. Okay. Now you can actually clone out of Genie. One thing I've noticed, I'm not sure why this is, but if you try to minimize these Java applications, they just, no, they don't let you do it. I don't really know why that is. So we can actually close Genie. I don't want to save the session, but you can if you want to. And now we find the text file, which is this gene report. So that we can now open in Excel. Xiaoyun. What's a hit and what's not a hit? No, so rigor does not do that. It's assuming that you've got a continuous list of scores and that all throughout that distribution, the numbers are meaningful, right? Which we know isn't always going to be true, but that's the sort of thing you need to, this is going to be specific for your screen. And some screens, probably it is relatively meaningful. The amount of change in the abundance of a hairpin is meaningful from top to bottom of the list. If you're doing a positive selection screen where you get your sequencing back and you realize, okay, there are 5 hairpins that scored and the rest are all zeros, you'll still get a number for those zeros. They'll still have a rank, but that rank is totally meaningless, right? So there is definitely a level of you want to go back and look at your data and make sure, okay, does my hit list match up with what my intuition of how far into a hit list I should go? Absolutely. Dave, do you want to comment further on that?
Yeah, I'll just, I don't know if we reiterate the question. Are there any thresholds baked into this analysis? And there aren't. As John said, it gives you a continuous list for every gene, but we would assume that only the top of the list is actually going to have any power and be meaningful. But it gives you a ranking for everything. Now,
right, it's a ranked list of, what you get at the end of rigor is a ranked list of genes, but I think what we're trying to say is that how far down into that list there's meaning will vary wildly based on the screen that you did. And that's something you should always be attuned to. Like, okay, I did a screen and I found 5 hairpins at the end. Okay, you shouldn't have a hit list that has 50 genes in it because you only had 5 hairpins.
Right, and it's harping on the same thing, but it's very important. The genes at the very extreme in the list, you have some faith in if your screen had power. But when you get down to the middle of the list, if you screened 15,000 genes, the 7,500th gene you wouldn't expect to be any different than the 8,500th gene. They were both inactive.
And that's a general point about this analysis. I mean, it's very easy to, once you get into spreadsheet land, to become detached from what happened in your experiment, because things always get a list, things always get a number, and it's very easy to become detached from, well, okay, what actually happened in my screen? Where's their power? Where's their not? Where's their noise? Where's their signal? So always keeping that in mind when you do these things, because it's very easy to get lost once you're in spreadsheet land. Okay, so here's the output of rigor. It's a file that looks like this, and there's a few columns in here. First, there's the gene. There's the hairpins that target the gene. If I click on it, you can see up in the formula bar that it's the sequence for all the hairpins all the various hairpins that went into that targeted this gene. It tells you how many different hairpins there were for a gene. It tells you the ranks of all those hairpins, and they are maintained in order, meaning the 10,953rd hairpin had this first sequence. It tells you the normalized enrichment score and the gene rank. This is what we actually care about. So here we can sort by gene rank. And you can see that in this particular screen, the number 1 scoring gene was MYL12B. The number 2 scoring gene was MYH9. You also get a p-value and a p-value rank. The p-value will obviously relate to the number of permutations you did, how small your p-values can be. If you only did 1,000 permutations, you can only get a certain p-value. You'll notice that the gene rank and the p-value rank, they're not identical, but they do track quite well as you scroll on down, which is totally what you'd expect. It's just there's slightly different ways of calculating. I always use the gene rank determined from normalized enrichment score, but that's not gonna push your results one way or the other. But I prefer to use the gene rank. But you also get a p-value and a p-value rank. These are just descriptive fields. They might or might not be meaningful based on your screen. So there's the number of hairpins that scored in the top 500, the number in the top 1,000, the number in the top 5,000, etc. Sometimes that's meaningful, sometimes it's not, but it's just a report that comes with it for free. To be honest, when analyzing screens, I never really use these fields to decide anything, but they're there for your enjoyment. Okay, so now you have a gene rank. Okay, now if your screen was really just one final list of numbers, then this is more or less your hit list. Now you could go back and try it with, instead of weighted sum, do second best. Now the genes will line up very nicely when you do that. If you want to, if you had multiple different conditions, or let's say you did the screen in multiple different cell lines, you'll get a gene list from cell line 1, get a gene list from cell line 2, and then either use an AND operator and take something that's scored in all cell lines, or take the OR operator and take something that's scored in any cell line. How far down into your hit list you should go? Sometimes you can get that from the screen that you did. Again, in the case of coming back to the strong positive selection, if very few things scored, then you shouldn't take 1,000 genes as your hit list. But for a lot of screens, it's not obvious to say, there's no bimodality between, this is the signal, this is the noise, and I know exactly where to draw the cutoff. So practically what tends to happen is, well, what's your next assay, and how many things can you do in your next assay? It's a very practical but a very useful way to say, okay, here's my hit list, or this is what I'm going to follow up on first. Okay. Yeah, absolutely.
What is the normalized enrichment score? That's the weight either your second best or.
Yeah, the normalized enrichment score, it's weighted by your second best, and Dave, I can't do a good job of explaining NES, can you?
Well, there's the weighted sum is this that you talked about, is just weighting the rank of the second best hairpin, 75% of the score, and the first best hairpin, 25% of the score. In terms of the normalized enrichment score, that's actually used in the KS analysis that John mentioned earlier. It's the amount of enrichment for that gene's hairpins versus the whole set using a certain metric.
Well, actually,
I don't know how it plays in here.
Well, when we get to GSEA analysis, I'll be able to illustrate what an enrichment score is visually in GSEA. So for now, why don't we hold off on NES, because here it's a little more, I don't have good graphics to visualize what it is, but when we get to GSEA, then you can see it a little bit better.
Here, whenever we do weight or something, you only use top 2 ascertain.
That's right.
Here.
That's right. That's right. So yes, it's an NES, but it's still only using the top 2 hairpins. That's exactly right. Okay, so if this is all you wanted to do, then you could generate your hit list from this. But one question, and I'm not saying you need to go further than this. I mean, if this is, you did a particular screen and you see genes that you're done. Okay, here's your hit list. Go. I will say that people sometimes spend far too long agonizing over their hit list. They want to analyze it every which way to Sunday. If when you're analyzing your hit list, you find that the method you're using wildly changes your results, that's not a good sign. Now, I'm not saying that different results will all give you, or different methods of analysis will all point to exactly the same thing, but they shouldn't be wildly different. But one question that often comes up is, okay, I want to move past the gene level analysis, and I want to say, are there any gene sets that are enriched? I want to have some sense of networks or pathways. So how do you do that? One way to do that is through an approach called gene set enrichment analysis, or GSEA. Is developed by Arvind Subramanian, among others. Arvind still lives up on the 4th floor. And it's a way of taking a list of genes and saying, is there something coherent in this list? I can't look at the gene symbols and say, aha, these are all mitochondrial proteins, or these are all NF-kappa B signaling pathway proteins. You want to do that in an unbiased way, and that's what gene set enrichment analysis lets you do. So there's a program to do that. Let's go look at that. So that is, let's see, GSEA. Here we go. So you can log into GSEA, which you don't even need a password. Well, you need to get a password first, but you just log in with your email address. And then you can download the Java implementation, okay, which if you want to follow along, hopefully you've already done. But it's a website, broadinstitute.org/GSEA, and it's freely available software. So we launch. Oh, well, let me launch it first, actually. Sometimes it takes a little while to load up. Okay, so we launched GSEA. Good. GSEA was designed and is still most heavily used for analyzing expression signatures. So you generate an expression signature from your cells / perturbation / whatever of interest, and you run an Affymetrix array. And then you want to compare the output of that array to some pre-existing microarray data. That's what it was originally designed for. So using GSEA for gene lists requires a bit of a hack. I mean, a supported hack, but a bit of a hack from what GSEA was originally designed to do. So this might seem a little convoluted, but again, the reason is that this is not particularly what GSEA was designed to do, to take gene lists from hairpin screens and get enrichment. Because of that, the hack means that practically your hits have to be the highest number. Okay, so we want the exact opposite of this gene rank. Okay, GSEA would look at MYL12B and say this is the worst gene in the screen, put it at the bottom of the list. It's pre-baked that your hits have to be high and your non-hits have to be low. So there's a lot of ways to do that. One way of doing it is just to rank in the opposite. So here there are, let's see, how many, oops, 15,275. So 15,275, and we'll do -1. And copy that all the way down. There are certainly more sophisticated ways of doing it. Just for the purposes of demonstration, we'll stay with this means of doing it. Okay, so we now, and also the input can only have two things, gene symbol and number, and that's it. So we'll delete everything else. We also need to delete our headers. Okay, so now we have this list, again, where the hits are the highest numbers, the non-hits are the lowest numbers. Now, this is the part that is not obvious at all, but we want to save this as a tab-delimited text file, and I'll just label it GSEA input.rnk. It has to be a .rnk file. If you try to load it in as a .text file, GSEA won't know what it is, and you won't be able to run it. So you have to save it as format tab-delimited text, but name .rank. Okay? Now, Excel will tell you you're going to lose features. Yep, that's fine. And then we can close Excel for now. Okay. And again, you can't minimize that. But OK, now if I go to my desktop and I look at GSEA input.rank, it looks fine. It's a .rank file. You are wrong. If you, at least on a Mac, if you go to Command-I and look at the information for this gene, you will see that it's actually a .rank.text. So you need to now edit it down to just being a .rank. Excel will always append that stupid .text on the end. You need to remove that. In a Windows environment, I don't know if you necessarily have to do this. I think it's easier to see in the actual file name. But in a Mac, you have to open up this window, delete the .text, and change it back to only a .rank. Okay? And then it says, do you really want to do that? Yes, I do. OK, so now we can go back to GSEA. Okay? And again, just like GSEA, there's a whole world around it that I'm not going to go into details at all. There's plenty to read about. Both papers and the help section on the website is quite extensive, very useful. With any tool, whether it's Genie, GSEA, or any other tool you want to use, don't just blindly use it. Learn what it's doing, because it's very easy to be led astray by things that the program lets you do, even though they're actually terrible ideas and will lead you astray in your analysis. So really understand every step in the analysis, what it's doing, what the output means and doesn't mean. Otherwise, you can very easily be led astray. So know what you're doing. I mean, these software packages can very easily be black boxes, but you don't want your, when you go back to the bench to have been led astray. So we want to load data, and we'll browse for files. So on my desktop was this GSEA input.rank file, so I choose it. And it loaded it successfully, no errors. Okay. Notice how you kind of have to know how to do this. Like, supported file formats doesn't even mention .rank, so you kind of have to know that you can do this. Then you want to go up to Tools, and we want to run GSEA pre-ranked. You don't want to go over to run GSEA over there, because that's not the pre-ranked version. You want to go up to Tools, GSEA pre-ranked. Okay. Boop, boop, boop. that opens up this. Now you have a few choices. I should say that there are basic fields and advanced fields. The default settings, you don't need to change at all, and they don't particularly affect the analysis. So the basic fields and the advanced fields, it's a bit hard to see. Oh, if I scroll over here, you can show them. You don't need to change them. They're fine as is. So the first thing you got to select, well, I won't do it in order. If you had loaded multiple ranked lists, you would select which ranked list you want to run it on here. Since there's only one in there, it automatically selected GSEA input. Collapse data set to gene symbols, false. We already have done that, right? Rigor gave 1 score for every gene symbol. So we've already collapsed the data set to gene symbols. To be honest, this option shouldn't even be here. If you go to the help page for GSEA, and it explicitly discusses using the GSEA pre-ranked, it says set this to false. You do not want GSEA collapsing your hairpins to genes. It will do it in a random way, not having anything to do with your score. You can run the program and you'll get a result, but it will not have been done based anything at all on the score of your shRNA. It will have just randomly picked a particular hairpin to represent a gene and go from there. So true shouldn't even be an option in this particular menu. You absolutely want it to be false. Chip platforms. You want it to be gene symbol because that is what you used. That is what is designating the gene. That's how it knows, okay, this is what the gene is. In theory, you could use seq accession, but again, everything we've been doing has been in gene symbols. So you select gene symbol.chip. There's a whole bunch of others down here. And as you scroll through them, you can see that a lot of them are based on, again, Affymetrix data. But we want to use the gene symbol. Should also point out, this is important, this is all assuming you're human. If you're mouse, then you need to do some symbol conversion to work in mouse space, which makes some assumptions about how closely related is the mouse homolog to the human homolog. That's dicey. You can do it. I wouldn't hang my hat on it as much. It can be done, but you still need to convert the mouse to the human. So anyway, you want to be gene symbol. And then the last thing you got to pick is the gene sets database. And this is what gene sets do you want to query? And it'll pop up this menu. Now, this menu is a little confusing. First, there's a bunch that are bolded. Notice how 3.1, 3.1, these are all 3.1s. Those are bolded. Below them are previous curations of gene sets. So the version 3, the version 2.5, the version 2, the version 1. You don't want to use those. You want to they're increasingly inclusive, so there's no reason to not use anything but the latest. So you want to use version 3.1. You'll notice that they also have, at the end, various types of sets. So here's the positional one, but then curated, there's 1, 2, 3, 4, 5, 6 curated. Okay? If you select, now this first one, because it's labeled all, is all of these 5 things below it. Or you could select just one of them. What you can't do is select all and a subset of it. It'll crash if you do that. So you either need to select, so you generally, there's no reason not to select one of the alls, unless you only, for some reason, were interested in, I only want to query KEGG, or I only want to query microRNAs, or I only want to query biocarta. But in general, you query all. Now, in theory, you should be able to take, well, I don't want to run this 6 times. I want to run the positional database, the curated database, and I'm command clicking right now, the motif database, the computational database, the gene ontology database, and the oncogene signatures. So that's 6 databases. If you try to query them all at once, you will crash. I think you can do 2 at a time. I don't think I've ever been able to do 3 at a time. So we'll just run it on 1, though. So we'll just run it on the curated symbol set. But if you're doing your analysis, there's no reason you wouldn't want to query all 6 of these different types of gene sets over time. So you click on all, and then down here, you click on run. And it's running. You can see that down here it is running. Oh, hold on. Oh, I wanted to stop it. That's OK. The number of permutations, right now it's set at 1,000. Okay? So it'll take a while to run. They recommend, and I should have in this demo, change this number to 10 first, just to make sure that everything runs successfully. Then it'll only take a couple minutes to run. If it's running 1,000, then it'll actually take a little while to run. when you actually want to like get your final analysis, you want to run through 1,000 permutations so that the statistics you get out at the end are meaningful. You don't want to run on only 10 permutations. But to make sure that everything loaded in appropriately, you want to just first set it for 10 or something to make sure everything runs, and this changes to success at the end. So fortunately, this is why one comes prepared. OK, at the end, you'll get, yeah, let's pay attention to your slides, Doench. At the end, if you've done it right, you'll get a success. Oops. You'll get success down here. You'll click on success, and it will open up in your browser the GSEA report for the query that you just ran. Okay? And it'll tell you some various statistics, false discovery rates, p-values, et cetera. You want to click on snapshot of enrichment results. And when you click on snapshot of enrichment results, it'll open up a screen that looks like this. And these are the, they call them the mountain plots of the GSEA. And let's walk through. If you've seen these before, you can quickly look at them and decide if it's informative or not. But if you've never seen them before, it's nice to know what these things are actually looking like. So basically what it's doing is here are all the different, for a particular gene set, here's where each line represents a gene in that gene set. And what the program is doing is walking down your hit list, and every time it encounters a gene in a gene set, it increases in value. Every time it doesn't, it starts to drift back down. So the idea is if you shoot up very quickly, then that means you had a lot of lines, a lot of genes that are in this gene set were at the top of your hit list. Okay? So these are all ones where there is clearly an enrichment for genes in this gene set at the top of your hit list. So the value goes up and then starts to decay over time. Likewise, you can certainly have the case where a lot of hits are found on this side, where for whatever reason, that particular gene set was strongly depleted in your screen. So you can certainly see that too. For example, if we were to have run some Achilles data on a cell line and said, what genes are highly depleted in my list? And you'll find a lot of ribosomal genes, of course, are depleted. Ribosomal genes are essential, so a lot of them fall at the very bottom of your list of abundance in an Achilles screen. You can also click around and get reports for what are these lines, what are the genes. So in this particular set, which relates to cell migration, you can see some of these myosin genes scored that make a lot of sense. RNAi scores, that makes a lot of sense too. So you can see both what are the genes in the set and what are the genes in that set that scored in your hit list. Okay. Actually the DAPL. So that's what you do with GSEA. And again, it gives you a hypothesis that, okay, if I knew absolutely nothing about that list of those gene symbols, now I would have a sense that, oh, okay, so this is somehow involved in cell migration or growth cone collapse. You would probably want to go to the paper that generated these data and read about it, and if it was an area of biology that you knew nothing about, go to that and look at it. Sometimes you do this analysis and you're like, oh my God, I had no idea about this area of biology, and you really learned something. More often it's used to kind of validate the list itself, where you sort of know what the biology is that you're studying, and seeing genes related to that biology score are very encouraging. It's an unbiased way of saying, okay, you thought you were doing a screen related to glycolysis. Well, do you see glycolysis gene scores? So that's what you do with it. Another type of analysis to do is not with gene sets, but to use protein-protein interaction data. And this one I won't do live purposefully, but it's a DAPL algorithm that was developed by Lizzie Rawson and Mark Daly's group, among others. Same idea. So you can go to the DAPL website, and you can take your gene symbols, paste them into that field. And DAPL takes a long time to run, so you would definitely don't want to do it live. Also, you probably want to click yes on do you get the plot at the end, and do you want to color by p-value. But I tried to do it yesterday morning, and there were already 50 jobs running, so I couldn't even run it. So fortunately, I already had run one. You get a bunch of files, and again, don't just blindly use these. Go to the DAPL, it's been published. Go to the DAPL algorithm and figure out, well, what are all these things? What do these statistics mean? You don't just want to blindly use it. But for a nice visual, you get protein-protein interaction data. Okay, so you see okay, a lot of these genes, so MIWEL12B and MYH9, these have been known to interact. Okay, good. You know, a lot of the genes in this hit list that I pasted into that box are known to interact on the protein-protein level. So that's a good sign that your hit list is real, and it gives you a sense of, okay, if you need to maybe winnow down, well, maybe I should winnow down to the guys that interact with each other, and maybe this tells you about the biology. So in the final 5 minutes, I want to talk briefly about off-target effects in screening. Now, we know they're there, and a program we have written is called Meerkat to help you deal with this. There's other programs out there that they do basically the same thing. This is the one that we developed in-house. And this is because when we put our shRNAs into cells, they can do two things. They can do what we want to do, which is to get into RISC and bind to the gene of interest with perfect complementarity and cut it. But the other thing they can do is enter that microRNA pathway that I discussed way at the beginning this morning, bind to the 3' UTR of the gene with only 6 or 7 nucleotides of complementarity and induce translational repression of genes. So this seed sequence is defined as nucleotides 2 through 7 or 2 through 8 of the hairpin. So these are the LKO-based hairpins, and this is what they look like in red is after they've been processed by Dicer. So you want to look at your hit list. You know, are there certain seed sequences that pop up all the time in my hit list? Or are there certain UTRs of genes that seem to commonly be targets for the hairpins that scored in my screen? So here's one screening example of differentiation in AML cells. Basically, the postdoc sorted for hairpins that would turn down HOXB8 or otherwise cause the cells to differentiate and become green. So he did a pooled screen. All the facts look good. He got a hit list. HOXB8, the sort of control gene, scored as the #1 hit. Another hairpin scored as number 102. Things were looking great, but there were almost no other 2 hairpin hits in this screen. But we did see, geez, a lot of the hairpins that are scoring seem to have the same sequence motif appearing over and over. So we scanned across. We saw that, okay, this only occurs at certain positions. Enrichments for particular seeds only occurs at certain positions. So the seed sequence region definitely lit right up. You can then ask, does this match? Do these seeds match to known microRNAs? Are they mimicking known microRNAs? In this case, absolutely. So you can see that this sequence that appeared, 13 of the appearances of this seed sequence in the library, there are 19 times total this seed appears. 13 of them appeared in the hit list. And that matches the microRNA miR150, and there are other examples. Likewise, you can ask, are there any genes that a lot of these seeds tend to map to? And in this case, so what's plotted here is all the non-hit hairpins, how often they hit a UTR, and all the hit hairpins, how often they hit a UTR. And you can see this one gene, MIB, was a real outlier. 55% of the hit hairpins hit MIB, whereas only 20% of the non-hit hairpins hit MIB. So MIB scores as a gene. Now, sadly for this postdoc, this was all basically already worked out. The biology, it was known that miR150 targeted MIB and that MIB was important for development. But the idea is, you want to look for this in every single screen that you do. So one common question is, how common is this effect? So we looked at the Project Achilles data, and we just said, okay, when we do this same sort of analysis, how often do we see hairpin scoring, not because of the gene they're targeting, but because common seeds are showing up? And the answer is, it definitely occurs. So if you look across, like 200 Achilles lines, by chance, using the parameters we set up, you'd only expect about 0.2% of your hairpins to be enriched at this threshold. But you do see this, across all the various Achilles lines to varying degrees. Averages that about 10% of the hairpins that show up in your hit list, across the Achilles data at least, about 10% are seed-driven. But it's not like this occurs in every screen either. So here's 2 screens just put on the same distribution, looking at very similar phenotypes. So rescue from a drug. One screen, this effect did not occur at all. Another screen was way out here. 27% of the hairpins seem to be scoring because of this seed effect. So what should you do about this? Well, first, you should always look, right? They can be found in screens. They don't have to be found in screens. They can be found there. So you look for them, and you subtract them out if they're there. If you think that a hairpin is scoring because it's a seed, don't necessarily use it, or at least be cautious about using it to determine anything about what you think its on target will be. But you can also actually use it to discover microRNA-related biology. This screen, for example, this screen B, turns out this is a pretty novel microRNA involved in the biology of interest, and the postdoc doing that project is actively following up that microRNA and the targets of that microRNA. So certainly you can imagine off-target effects as bugs of the data, but you can certainly turn it into features so long as you do the appropriate analysis. And I should also point out, actually, that's one reason I include this slide. This is from analysis of Achilles data. It's not like these screens are rife with off-target effects, and thus there's no ability to look at on-target effects. If you take PI3 kinase mutant cell lines and ask, are the PI3 kinase hairpins specifically essential to those? Yes. BRAF, yes. KRAS, yes. NRAS, yes. mTOR, yes. It's not like the results of these screens are contaminated by these off-target effects if you do it right and require multiple hairpins targeting your gene of interest to score. So even though if you walk away from this saying, 10% of hits are wrong because of off-target effects, that's the wrong way to interpret this slide. This is that 10% on average of hairpins might be scoring from an off-target effect, but not 10% of genes. If you do your analysis right, then the number of genes that score to an off-target effect is very different and a much smaller number. So with that, I just want to point to where sort of analysis is headed. Yeah. Yeah.
So now that we, and we have good data to show, what the extent is, going back to our pools and mini, I wonder if looking back, if there's any modification, we should know exactly, right, what are the seeds in, let's say, the kinase or whatever, the pools that we have. And I wonder if there's a plan to change them or to just, upfront say what they are.
Yeah. So the question is, since we know that this seed effect occurs, what can we do about it proactively? I'm not sure there's too much we can do about it proactively in the sense of just eliminating hairpins. I mean, every hairpin has a seed. They're all going to match to something, so you can't really eliminate it. So that's where you want to be able to, again, just have multiple hairpins per gene. As it's such a simple rule, but it's amazing how often people quickly start to violate it and how quickly that can get you into trouble. So just because this also answers the question, where is analysis headed? So, right now, most forms of analysis map each shRNA to a gene, but not necessarily to a transcript. So we're ignoring splice isoform differences. So one thing we're definitely looking at and starting to incorporate in our analysis, not in a way that's toolified yet, but is mapping shRNAs to individual transcripts, not to individual genes. But also there's a lot of data. I've mentioned Achilles a lot here. The Achilles data came up with a great algorithm that was just very recently published called ATARIS that looks at how well the hairpins across all the massive Achilles data, how often they agree with each other, so to really get a sense of what's on target. That was developed by Aviad Cherniak, among others, up on the 4th floor. I already mentioned that we have this huge data set of qPCR data, so to get a sense for if it's on target, it's not likely, if a hairpin isn't knocking down its gene, then it's not likely to be an on-target hit. Dave mentioned the L1000 assay. If you've heard about it, it's a very large data set looking far more deeply than anyone ever has at how well shRNAs are working across many different cell lines in a very deep readout of gene expression. So one thing it's very much in R&D mode, not ready to use yet mode, but in R&D mode is to take L1000 data and come up with what really are the best hairpins for various genes. Does that change across cell context or not? But what can we do about this problem? So that's where analysis is headed.